Hi, this is Dr. Rosario, and this is our third and final lecture out of Chapter 8. In this lecture, I will be talking about enzymes. So we're going to start with what is the role of an enzyme? What is its purpose in metabolism? Then I will discuss the structure of our enzymes. And then finally, I'm going to get into the different things that can control or affect enzyme activity. So let's start with enzymes. The book definition of an enzyme is a biological catalyst. All right. What is a catalyst? Well, a catalyst is something that speeds up the rate of a chemical reaction. So enzymes are molecules that speed up the rate of a reaction within our cells and within our organisms. Remember, our metabolic reactions often occur in pathways, taking us from our reactants that are our starting molecules towards our products. These pathways, as we move through them, each step, each reaction within the pathway is controlled by an enzyme. All right, so if enzymes speed up the rate of a reaction, let's discuss what determines reaction rate. So what does not determine reaction rate is delta G. Delta G tells us whether or not a reaction is spontaneous or not, but it does not determine the reaction rate. Right? Remember, delta G is that change in free energy. The reaction rate, the speed of a reaction, is determined by something called activation energy. So now we need to define that for you. So here I have a figure for you that's an example of an exergonic reaction where it has a negative delta G, right? It's energetically favorable. You can see energy is being released. And this is a hydrolysis reaction, right? So, of course, many of our catabolic reactions are hydrolysis reactions because those are the chemical reactions that are used to break down our polymers, right? By adding a molecule of water. So in this reaction, we have two monomers that are held together by a bond, and then we have our molecule of water. Those are going to be our reactants. We're going to break the bond between our two monomers, and we're going to break the bond between the hydrogen atom and the hydroxyl group within the water molecule. So we're going to be rearranging atoms as this chemical reaction proceeds. Now again, this reaction is energetically favorable. If we were to trace the energy of this reaction over the progress of the reaction as we move from reactants to products, the reactants have more energy than the products, right? Meaning our delta G is negative, right? Because delta G is the difference between the energy of the products and the energy of the reactants. This does not tell us the speed of the reaction, however. It just tells us that the reaction is energetically favorable. In this reaction, as we move from reactants to products, we are going to move through something we call the transition state. In the transition state, we have those bonds that are going to be broken in this reaction and the atoms that are going to be rearranged. They're forced into a position that makes the reaction possible. This in-between state where we're no longer the reactants, but we haven't quite yet formed products, is called the transition state. In order to get to the transition state, to distort the molecules in just the right way, we have to put in some energy. That energy is the activation energy. Now that energy that we put in, we get back out as the molecule moves from the transition state to the products. But that transition state is a higher energy state than what we have in our reactants. So let's put this on our graph that we just had. So here we have our reactants, we have our products, but we have to pass through the transition state. So we're going to initially invest some energy in, and then the energy will be released. Okay. Now, notice that even though the transition state is higher, right, and we have to put some energy in, that energy is released back once we get past the transition state. So the activation energy, which is the amount of energy necessary to get to the transition state, does not affect delta G. Activation energy affects the rate of a reaction. It is what determines how quickly a reaction can proceed. Okay, so the activation energy is the energy necessary to reach the transition state. It does not affect the delta G. It does not determine whether a reaction is exergonic or endergonic. It simply determines the rate of the reaction. Now, activation energy, like I said, it does not determine delta G. You can have a reaction that is incredibly energetically favorable. A great example of that is the oxidation of glucose. Right? So the oxidation of glucose releases a lot of energy. Wood, right? Wood is made of what biological molecule? Do you remember? Cellulose, right? And cellulose is chains of glucose. Okay, so when we break down glucose into carbon dioxide, right, by reacting that glucose with oxygen, and we release carbon dioxide and water, right? That's what we do when we burn a log. We are oxidizing the glucose in that log. So, but you can take a log and you can set that in your fireplace, and if you don't do anything to it, you just set it there, the log does not spontaneously combust. That reaction does not proceed, right? Because in order for this reaction to occur, we have to overcome the activation energy. The activation energy is so high that under standard conditions, right, your log is not going to break down, right? Your glucose is not going to break down. So what do? We take a match and we light that log on fire, right? By using that match, what we're doing is we're introducing a little bit of heat. Heat is a form of energy, right? That provides the energy necessary to overcome activation energy. Now, within cells and within living things, heat isn't particularly useful for overcoming activation energy because bodies are maintained at a relatively constant temperature. So instead, organisms use enzymes to speed up the rate of a reaction. Most of the chemical reactions that occur within cells have such a high activation energy that they would not occur at a rate that could sustain life. So enzymes allow for these reactions to occur. So a great example of this, if we go back to the oxidation of glucose to carbon dioxide, that process, that same process that happens when we burn wood, it happens within each one of our cells in what's called aerobic respiration. And we're going to talk a lot about respiration in the next chapter, but what I want you to see here is we're going from glucose to carbon dioxide. Glucose has 6 carbons. We're going to take those 6 carbons and convert them to carbon dioxide in this process. Notice all the little arrows as we're going through glucose catabolism. Each one of those arrows represents a different chemical reaction within this pathway. And each one of those reactions is controlled by a different enzyme. Now, in the cell, rather than just burning the glucose and releasing all that energy as heat, instead the energy is captured a little bit at a time. And that's really what we're going to get into in the next chapter, is how cells capture the energy of glucose. But a little bit is captured at a time so that then we can use that energy to sustain life. So enzymes allow all of this to happen. So enzymes, like we said, they are biological catalysts. They allow for the reactions that support life to occur at a rate that is fast enough to support living things. They are usually made of protein. Now, not all enzymes are made out of protein. Some enzymes are made out of RNA, and they're called ribozymes. We'll discuss the ribozymes as we get towards the end of the semester when we talk about transcription and translation. Enzymes, like all catalysts, they lower activation energy. And by lowering activation energy, they speed up our biochemical reactions. But they do not alter the delta G. They do not alter the energy, the overall energy change of the reaction. So enzymes lower activation energy, but how do they do it? If heat can if heat can speed up a reaction by simply adding more energy to our reactants, how does an enzyme lower activation energy? How does it speed up a reaction? Well, for one, enzymes can bring reactants together in physical space. So in three-dimensional space, the enzymes can position molecules so that they're more likely to interact with one another. Enzymes can bind to reactants and distort the reactants, stabilizing the transition state. Enzymes can provide a microenvironment where a reaction can occur that is separate from the cytoplasm of the cell. Some enzymes can actually directly participate in a chemical reaction. So the side chains of the amino acids that make up the enzyme may bind temporarily to a molecule and form a chemical bond, but then it's important to remember that the enzyme is not permanently altered in the process. Ultimately, the enzyme is released the same way that it started, and it's not permanently altered or destroyed in the process. So the structure of an enzyme, like I said, enzymes are made primarily of protein. And if you learned anything so far this semester, you know that the structure of a protein, the shape of a protein, determines its function, right? That three-dimensional shape, that what we call tertiary structure of our protein. So here we have an enzyme pictured for you, and it's a space-filling model so you can get kind of an idea of its three-dimensional shape. Now, every enzyme has a site where the reaction actually occurs. That is called the active site. And the reactants, they bind to that active site. In an enzyme catalyzed reaction, the reactants are now called substrates. All right? And you'll notice in this picture that the substrate is complementary to the enzyme, right? They match one another, kind of like a lock and a key match one another. Their two shapes are complementary. Some enzymes require additional molecules for their activity. So a cofactor is an inorganic substance that is necessary for the activity of an enzyme. And on the left-hand side of your figure, we have an example of what's called a DNA binding protein, or a zinc finger domain. These are proteins that bind to DNA. Now, in order for these proteins to be able to bind to DNA and interact with that DNA, they have to have zinc as part of the molecule. Zinc ions are positively charged. DNA is negatively charged, so it enhances the interaction between the enzyme and its substrate. Zinc is an example of a cofactor. It's an inorganic substance that is necessary for the activity of an enzyme. A coenzyme, on the other hand, is an organic substance that is necessary for the function of an enzyme. So for example, hemoglobin is a protein that binds and carries oxygen. Hemoglobin has heme groups present in the protein that allow it to bind to that oxygen. The function of that protein depends on that coenzyme, right? Coenzymes are organic cofactors. All right, so proteins are primarily composed of protein. They may have cofactors or coenzymes associated with them. The function of an enzyme is determined on its interaction with that substrate. So we have what's called the induced fit model of enzyme function, where when the substrate binds to the enzyme at the active site, the active binding itself alters the shape of the enzyme. The enzyme ends up kind of closing in on that substrate because the electrons that are part of the substrate and the electrons that are part of the enzyme, they get shifted around through that molecular complementarity as those two molecules interact. So you have what's now called the enzyme substrate complex. The enzyme has bound to the substrate and it's closed in around that substrate. That interaction can then distort the reactants, distort that substrate, and then push the reaction towards the formation of the products by lowering the activation energy, stabilizing the transition state. So this is called the induced fit model of enzyme function. So we have what we call the catalytic cycle, where you have substrates, right? And you can have one, you can have two, it depends on the enzyme, you can have three or more even. The substrates bind to the enzyme at the active site. Once you have the binding at the active site that distorts the shape of the enzyme, you get that induced fit that then in turn lowers the activation energy. It allows for the reaction to proceed quickly, and then once the products are formed, they are released from the enzyme. What you end up with at the end of this cycle is that enzyme is ready for another set of substrates to bind to it. So you can have the cycle occurring over and over and over again. One enzyme can catalyze thousands of chemical reactions. All right, so now since an enzyme is composed of protein, the things that affect protein shape are going to affect the function of an enzyme, right? So one thing that can affect enzyme activity is temperature, right? So most of the enzymes in our body function best at 37 degrees Celsius because our enzymes, their shape is maintained at that 37 degrees Celsius. However, organisms that are adapted to living in warmer environments, like a thermophile, a microorganism that lives in a hot spring, their enzymes may function best at a much hotter temperature, like 75 degrees Celsius. What I want you to see though on this figure, right, you have different optimal temperatures for these two different enzymes, but there's one important thing I want you to see on this figure. Notice that as we increase the temperature, the rate of the reaction steadily increases. Because again, anytime you increase temperature, that temperature can be used to overcome that activation energy barrier. However, once you reach a certain temperature, that reaction that's being catalyzed by an enzyme, the enzyme itself, remember it's three-dimensional and it's made of protein, the shape of the protein can get distorted. So in the case of human enzymes, once we get above 37 degrees Celsius and really up above 40 degrees Celsius, our enzymes completely lose their shape. And you'll notice that the activity of a enzyme catalyzed reaction completely disappears. Right? It drops off because we've denatured the proteins. They're no longer active. The same holds true for our thermophilic bacteria. It just happens at a higher temperature because these proteins tend to be more stable at those higher temperatures. So the environment, the temperature within the environment affects the activity of an enzyme. Another important thing that's going to affect our enzymes is going to be the pH of the environment. Again, proteins, their shape is influenced by the pH of the solution in which they exist, right? So most of the proteins within our body and within the cytoplasm of our cells are going to function best at a neutral pH at about 7.2. That they hold their shape at that pH. Well, pepsin and trypsin are two exceptions to this rule. Pepsin is an enzyme that is found in the stomach, where the pH is very, very low, and its enzymatic function is going to be highest at that very low pH. It's going to have just the right shape at that pH for it to be able to catalyze the reaction that breaks down the proteins that we eat. Trypsin, on the other hand, which is found in the intestines, where there's a more alkaline pH, it functions best at a higher pH, at about a pH of 8. And again, it's doing the same thing. It's breaking down the proteins in our food. So these two enzymes are examples of where the pH that these enzymes are sitting in is going to affect the activity. If they are within an environment that is not within that optimal pH range, they can't function at all, right? Because the pH alters the shape of your given proteins. Right, other things that affect enzyme activity are inhibitors. So here on the right-hand side of your screen, you can see my substrate and it's binding to the active site. You can see that the active site is complementary to the substrate. Inhibitors come in a couple different forms. Inhibitor is a molecule that prevents that enzyme catalyzed reaction from occurring. A competitive inhibitor is a molecule that binds to the active site of an enzyme instead of the substrate, right? So usually our competitive inhibitors are very similar in shape to our substrates. Many of the drugs that we use in medicine are competitive inhibitors of enzymes. A great example of this is ibuprofen. Ibuprofen inhibits an enzyme called cyclooxygenase. That drug is a competitive inhibitor of that enzyme. A non-competitive inhibitor, on the other hand, is a substance that binds to an enzyme at a location other than the active site. Sometimes we call that the allosteric site. And the reason we call it the allosteric site, allosteri means to change shape. So in non-competitive inhibition, that molecule binds to the enzyme, it changes the shape of the enzyme, and changes the structure of the active site without interacting directly with the active site. So that's non-competitive inhibition. All right, there's also ways in which the cell itself can control enzymes. One way is to control the amount of enzyme present. Another way is what we call allosteric regulation, feedback inhibition, and then finally localization. So the amount of an enzyme. We can, as a cell, regulate the amount of protein present in that cell a couple of different ways, all right? One is through simply regulating gene expression, right? How much RNA are we going to make from a given piece of DNA that can then be used to make protein, right? So that's what we call regulation of expression. We're regulating transcription and translation of the gene that makes a particular enzyme. Another way we can regulate the amount of enzyme is by regulating the stability of that enzyme. So if a cell does not need a given enzyme anymore, it's going to break it down, right? Some enzymes have a very long half-life. They can exist within the cytoplasm of the cell for a very long time. Others are degraded almost immediately after they're made so that they don't stick around for too long. So again, cells can control which reactions are being used by the cell by controlling how much of a given enzyme is present. Right, another way that enzymes are controlled is through what's called allosteric regulation. So I mentioned the word allostery earlier. Allostery means to change the shape of a protein. Now you can have allosteric activation and you can have allosteric inhibition. Allosteric activation is where you have a substance, we call it the activator, that binds to an enzyme and it stabilizes the enzyme in its active form. On the other hand, in allosteric inhibition, you have a molecule that can bind to the enzyme at a site other than the active site, when then that stabilizes the inactive form of an enzyme. Again, that's going to be your non-competitive inhibition, right? Allosteric inhibition is a form of non-competitive inhibition. Another type of allosteric activation is what's called cooperativity. So in cooperativity, what happens is you might have a multi-subunit enzyme that has, let's say, 4 different polypeptides. Each polypeptide is made up of an enzyme and then they are linked together, all right, through quaternary structure. The first subunit, when it binds to the substrate, that in turn stabilizes the other subunits, making it so that they're more likely to bind the substrates when they interact with them. We see cooperativity in our hemoglobin, for example, because when the hemoglobin molecule with its 4 different subunits binds the first oxygen molecule, it then enhances its ability to bind to other oxygen molecules. So that's an example of cooperativity. Another type of regulation of enzymes is something called feedback inhibition. So in feedback inhibition, this also is somewhat a form of allosteric inhibition, but it occurs when the final product of a long pathway inhibits the first enzyme in the pathway. So this example that you have on your screen, we have the production of an amino acid, isoleucine. And you can see that you start out with the initial molecule is threonine, and through a series of chemical reactions, we convert threonine to isoleucine. And there's 5 different enzymes in this pathway. Now, in feedback inhibition, what happens is as that end product builds up, and if there's too much, the cell can turn off that entire metabolic pathway. The way it does that is the isoleucine, the product of this pathway, is an allosteric inhibitor of the first enzyme in the pathway. It's kind of like when you have a factory, right? And I love to use the example of chocolate bunnies, right? If you have a factory that is producing chocolate bunnies and you start making too many chocolate bunnies, you need to shut the factory down. But you don't just stop it, right? You turn the first machine off and then you allow for the other machines to continue forward so that you finish that run, right? You don't have a bunch of partially made chocolate bunnies. That's essentially what happens when we have feedback inhibition. The cell is sensing that it has enough of a given product in a pathway, and it uses that excess to turn off the first enzyme in the pathway. And then the last way that enzymes can be regulated is through something called localization. And we've actually talked about this a little bit before. So remember your lysosomes? Your lysosomes are those digestive components of your cell, right? They're organelles that are specialized for breaking down biological molecules. Your lysosomes serve as compartments where certain enzymes can be separated from the rest of the cell. So our acid hydrolases, our nucleases, our proteases, they are separate from the cytoplasm of the cell, and they're only active at acidic pH, right? Whereas the cytoplasm of the cell is closer to 7.2, your lysosome has a pH of about 5. By compartmentalizing these enzymes, by localizing them to a particular spot within the cell or within the body, we then are regulating the enzyme activity. We're keeping it localized to a very specific location. Okay, so that is it for this particular lecture. We've talked about what is an enzyme, right? It lowers the activation energy of a reaction. It is generally composed of protein, and anything that can affect that protein shape is going to affect the activity of that enzyme, and then affect the metabolic pathways controlled by those enzymes. All right, as we move into chapter 9, we're going to get into the details of glucose catabolism. This is really at the heart of all living things, this ability to take the energy stored in glucose and release it to be used by the cell.